To cap off our discussion on carbohydrates, I think it's necessary for me to give some of the more popular uses, coupled off with some specialized uses close to me, though I don't want to get carried away by explaining too much. So let's start with the energy, perhaps the universal known use of carbohydrates. And of course, for us, glucose is the main sugar for energy. In the first place, it's a monosaccharide, so once it's in our blood and we need energy right now, it can be easily broken down via some metabolic pathways, and tada, we have our energy to do what we need right now. So we can pretty much say that the monosaccharide glucose is our immediate source of energy. Without it, we won't be able to do almost every single thing that our body can do. However, in some cases that we have excess glucose, like for example, you ate a lot during lunch and your body doesn't know where to store the glucose and it can't immediately be removed from your body, it can be stored in your muscles or your liver as glycogen, which is basically just thousands of molecules of glucose connected together via glycosidic bonds. In plants and probably some species of protists or bacteria, starch is the name of the glucose polymer, and in fact, starch and glycogen are not that far in terms of structure. So again, these are storage forms of energy, only used when the body or the organism deems it's necessary. Now, we also have structural carbohydrates, and I think for even high school level biology textbooks, it's always been mentioned that the cell walls of a lot of organisms are made of some kind of carbohydrate. Like for plants, I think a lot of us would be familiar already with cellulose. And for fungi, they do have cell walls also, but this time it's made up of the polysaccharide chitin. So as you can see, cell walls are always made up of polysaccharides, all right? They have to be big or long molecules in the first place. And then for bacteria, we have something called, I'll just throw in the generic name, peptidoglycan. Generic because this word only means peptide and glycan, but there are actually specific amino acids that make up this peptide portion or specific sugars that make up this glycan portion, but we don't want to discuss too much of that here. If you want, you can proceed to your microbiology discussions for more detail. For the exoskeletons of some sea creatures like shrimps or crabs, we also have actually chitin. So that's weird that fungi, which are a lot of them are microscopic, their cells are made up of chitin, and then all of a sudden you think that animals like crabs are so different from fungi, but it's also made up of chitin, their shells, I mean. So that's variety for you. Signaling, on the other hand, another use, is quite a complicated word because it basically covers all ways of organelles within the cell communicating with each other and also communicating with the external environment, which is actually a really, really wide area of things. So, for examples of what signaling means, we would gradually learn more about it as we have more examples. Like, for example, receptors. Receptors are things that we usually study in pharmacology. Let's say that I'm drawing here an example of a protein that if we can assume a drug would be able to alter the function, it would result to some beneficial effect in our body. Then let's say this is a drug molecule. So in basic pharmacology, what we usually assume is that when a drug fits here very well into a certain cavity of a certain receptor, depending on how well the fit is done or the fit is here, the effect would be accordingly strong. If it doesn't fit well, the medicine won't do a lot. And actually, one of the most overlooked things in basic pharmacology is that there are some carbohydrates, oligosaccharides, that actually modulate that. They control how well or how bad a drug molecule can fit into some receptors. So that's something that we could have as an example of signaling. Another very different way of signaling is protein folding. Now, hopefully you would remember in high school biology that there's this thing called the ribosome that creates proteins. So let's say that we have a newly made protein. Usually, a newly made protein is just like a strand, but it has to undergo a process called folding in order to become more complicated in shape. And usually, when it becomes complicated in shape, that's the time that it becomes functional. And in some cases, the cell would have to add various sugar components to a newly made protein in order for this protein to fold properly. Because if this does not happen, then we won't have a functional protein. It's like the ribosome made a protein for nothing. So that's another example of signaling. Another one is the ABO blood type, which is, I think, the blood type that we are all familiar with. Usually, when we are asked, right, in the hospital, what's your blood type? It can only be one of the four main answers: A, blood type B, blood type O, or blood type AB. Of course, there are more blood types, but we can zoom in on these three first. So the thing is, if you can imagine that this is a red blood cell, the surface of a red blood cell actually has oligosaccharide components, which is actually what would dictate, in terms of what would dictate, I mean, the blood type of that person. So, for example, if I have, for example, you can imagine that these are a series of sugars. This would be blood type O. Then, if you can imagine the exact same set of sugars in blood type O attached to the surface of a red blood cell, then you attach one unique sugar, then we have, all of a sudden, blood type A. So this is the only difference of a person with blood type A to a person with the blood type O. And in fact, if I have a blood type B, it's basically like blood type O again, but I have one extra sugar that's absent in blood type O, but for sure is different compared to blood type A. So those tiny differences in the sugar composition in the surface of the red blood cell would dictate the blood type of a person, at least for the ABO blood type system. Now, for the pharmaceutic and pharmacologic examples, I'll just really, brush through them. Some of the carbohydrates or carbohydrate derivatives are used as sweeteners, not just in, let's say, chewable tablets, but also some other cosmetics, things you can find in the bathroom like toothpastes and even food like chewing gums. I could think of something like sorbitol, okay? Diluents, which are basically things that you add to, let's say, a tablet in order to increase its size so that it could be taken more easily. Would you believe it or not, lactose, the so-called milk sugar, is actually one of the most common diluents. And then disintegrants are things that we add to tablets usually so that when a tablet is taken in by the body and it's in our gastrointestinal tract, the tablet would start to break apart. Without proper disintegration, then the tablet will exit our body as a tablet. So I think one of the most common disintegrants that's also a carbohydrate derivative is a starch derivative, sodium starch glycolate. But of course, there are more examples. For pharmacologic, I just thought of these four because some of them are well discussed in pharmacology, basic pharmacology, and some of them I see a lot during my internship, honestly. Mannitol is one of the more common so-called diuretics, osmotic diuretics, that are used for reasons that are not exactly the same as you would say for other diuretics like furosemide or others. For lactulose, it's a laxative, and in fact, one of the most popular laxatives of all time. Sucralfate, which is a derivative of sucrose with some sulfate additions, is actually what we call a cytoprotectant, which is just a word that we call for an anti-ulcer drug, which works by coating the stomach, preventing excess acid from injuring it. And heparin is one of the most well-known, if not the most well-known, intravenous or injected sorry, it's not a coagulant, it's an anticoagulant. Okay, so that's supposed to be it, but I think just to connect our carbohydrates to the next three biomolecules, I would just want to emphasize that carbohydrates are expected to join with some of our biomolecules, and you would have to welcome our carbohydrates again in the future. For example, there are actually combinations of proteins with sugars that we call glycoproteins. Well, actually, since I mentioned a while ago that some proteins are, embedded with sugars to fold properly, then I think you can call this a glycoprotein. Then there are also what we call glycolipids, which are lipids with sugars. And I think most of the glycolipids that we would discuss in the lipids discussion or portion are the sphingolipids. And for nucleic acids, I need to emphasize that without sugars, there wouldn't be nucleic acids. I think for laymen, The two nucleic acids are well known, the DNA and RNA molecules, and those first letters mean a lot because D in DNA would probably emphasize or most likely emphasize the word deoxyribose, while the R in RNA would emphasize ribose, which I hope you are familiar. Two of the pentoses we have used as examples before. So that would be it, and it's time to move to the next biomolecules.